everyone. My name is Pinar. I'm a postdoc here at the Schmidt Center, and I will talk about our efforts to use deep learning to identify regulatory interactions that are driving cell state transitions across the different regions of the trajectories, different differentiation trajectories, and so on. So many of the biological systems that a lot of us are interested in, and many of the biological systems that we see in nature are characterized by dynamic cell state transitions, where cells move between different functional identities, like embryonic stem cell differentiation and development, or the opposite of it, where with cellular reprogramming, we have the somatic cells being reprogrammed into stem-like state. Or it also happens daily in our bodies with T cell maturation and aging and so on. So of course, there is an underlying dynamical process that governs all of these cell state transitions, and these are driven by gene regulatory interactions. So these are non-static, they're contextual, and they also operate at different scales. For some interactions, effects may be instantaneous, and for others, there are delays. So some of the questions of interest when studying these dynamical systems is, why do cells that look initially pretty similar in terms of their genomic profiles end up going into different cell fates? And what are the regulatory interactions that are driving these across different scales of biological time? And once we have insights into these, we could get an idea about how to intervene in these systems so that we can reroute the trajectories. So of course, the fact that single-cell sequencing is destructive makes this challenging because we can't track the cells across time. What we have usually is just snapshots from subsets of the population at different time points. But since the sampling is random, we could actually very likely match cells across time points because with random sampling, we can assume that it's going to be a pretty good representation of the population at that time point. But now comes the challenge that at any given experimental time point, we have a heterogeneous mix of cell states. Because cell state transitions are asynchronous, they don't happen at the same time. So we get these cells across different regions of the landscape. This is actually both a blessing and a curse. It's a blessing because that means that with a limited number of, or with just a few snapshots, it's possible to cover a large part of the data manifold. So that makes the computational methods to infer trajectories possible. But it's also a curse because that means experimental time is not the same as biological time, and also because the progression speed changes along trajectories. So there are a lot of different approaches to understand drivers, because this is a very popular problem in computational biology, since a lot of systems are characterized by these dynamical changes. But a common theme is that a lot of them are identifying individual genes, and some of them are identifying drivers per trajectory and not the progression along the trajectory. But then the same transcription factors, for example, end up coming up multiple times in different processes or in different trajectories within the same population. So the question is, are they leading to different trajectories because of their different regulatory interactions, and can we capture that from the snapshots by understanding the dynamical process behind these cells? So our approach is to first learn a continuous time dynamics of these systems. We're going to use some flow-based models in order to parameterize the velocity field, and that's going to allow us to learn a time-dependent evolution of the of individual cells. And given these, we're going to, first of all, from the velocity field, we're actually going to try to infer a biological notion of time, and then along that time, we're going to use graph neural ODEs in order to understand gene regulatory dynamics. And the way we're defining a driver for interactions is that the interaction should both be strong. It could be negative or positive, but the strength should be high. and also the effect of the interaction. So if you perturb the target gene that's being upregulated, for example, if that makes a big difference in the velocity landscape, then combining these two together defines the notion of a driver for interaction. So of course, ideally, we would have multi-omic data, since that gives a lot more information into cell states and also the regulatory interactions, but we're going to start with transcriptomic data. Right, so what we have is samples from different time points, right? And what we're going to try to learn is a flow over these samples and the data distribution that the samples define. This is similar to the diffusion model that a previous speaker described, but in our case, we're not starting from a noise distribution because we already have data at t equals 0. So we're going to define mixture of Gaussians over our data points at multiple different time points. And we're also ignoring stochasticity for now. So given this, the velocity field that we learn basically defines a flow that takes a cell that's at a specific location at time 0 to a different location in the gene expression space at a later time point. And you want to learn these velocity fields such that it can explain the data that we see. And these velocity fields over individual samples also define a flow over the probability distributions. But we don't assume that this change in the gene expression is the only reason the gene expression will change. We will also incorporate a growth term. So you could compute these growth terms from prior data, as in annotations of apoptosis and proliferation genes, and that actually helps with convergence. But we're not going to keep it static, and we're going to learn it in the model as well. There's no unique solution to this setup because the continuity equation could be satisfied with multiple different velocity fields. So that requires us to make some assumptions on the velocity field. And we're going to basically penalize long paths and unnecessary mass destruction. So basically trying not to make any changes to gene expression or the density distribution unless we need to in order to explain the data. And this basically defines an optimal transport distance in the dynamic setting. And we also want to make sure that while we're trying to encourage shortest paths, the data flow actually happens on the manifold. And we are going to pre-train a VAE that basically uses a triplet loss to make sure relative distances in the neighborhood graph-based distances in the PCA space is also preserved in the VAE. Right. We continue to actually learn, update the VAE after the pre-training while jointly learning the velocity field because we found that this really helps with reconstruction and simulation of new data and also the sharpness of the partial derivatives that we're going to compute later on. And that's with both matching densities and also matching predictions observed to the observed data after reconstructing with VAEs. And there's just a word of caution. Because there is no correspondence between the predictions that we make and the cells that we see, we're going to use a Wasserstein distance, but Wasserstein distance is actually biased in the sense that your probability, the entropy of your marginals will influence the scale of the distance. So we use the biased version in order to make sure every batch has the same amount of influence over training. Right, so before moving forward, I just want to show you that using linear tracing dataset where we have barcoded cells that encode clonal relationships, we can actually check how well the trajectory inference happens. And if you look at all of the datasets, so what we have here is hematopoietic stem cells differentiating into neutrophils and monocytes. So we don't know which cell at the initial time point a cell from a final phase came from. But we do know that the progenitors, we can calculate basically the phase probability of progenitors using the clonal barcodes. Oh, this did not update. All right, my bad. if you look at the whole if you look at the whole data set, actually, I think it should be because this wasn't. If you look at the whole data set, we get about close to 99% accuracy. But the thing is, of course, making predictions along the differentiated, like the final fates, will be very easy. So that's not really a good notion of accuracy. The more difficult task is trying to figure out what the progenitors from the initial time points is for each of the cells at the end. And for that, we get close to 89% accuracy. Right, so this learns the generative model over the cell state transitions. But one thing that we mentioned is that we want to learn these interactions over a notion of biological time and not experimental time. But right now, the velocity field is taken in experimental time and is generating samples at a given experimental time. So in order to convert this into a biologically meaningful notion of time, we're going to decompose it into three components: gradient flow, rotational flow, and harmonic flow. The gradient flow has no curls at all. So this is basically cells being attracted to a specific point, and that describes the Weddington landscape, if you're familiar with the notion of marbles rolling down a hill to reach a final state along a potential landscape of differentiation. A rotational flow captures local cycles, so it's more related to cell cycle and stuff. For our questions, because we're interested in initial cells ending up in final fates, we're going to use the gradient flow decomposition. And what we do is basically order the cells on this landscape, and the landscape is not going to be a straight line, but this ordering is going to give us a notion of pseudotime. So then what we can do is create these trajectory-specific time series data, order them based on the landscape, and then train a graph neural ODE in the biological time, such that the changes in the gene expression of your regulators, of the regulators of a target, will try to predict the change in the gene expression of the target gene at that time point. For this, what we found is looking at expression itself is not very useful, but looking at the velocities ended up being more useful, and we were able to capture biologically meaningful relationships. And we add a time delay in order to also preserve interpretability. We're using assigned and linearized attention mechanism. I'm going to just highlight a few results, and then move on. So first of all, in iPSC trajectory, early on in the differentiation, I guess I should say reprogramming trajectory, we found that there's an interaction between FOXD and FOXO, and FOXD was previously validated experimentally to increase pluripotent reprogramming efficiency. On top of that, we also see some interactions that come up for multiple different trajectories, like MYC and KLF4 interaction, but what we see is the dynamic of when they influence the trajectory and how much they influence the trajectory actually changes. For iPSC trajectory, it becomes a highly influential interaction early on, goes down, but then comes up again during the iPSC when the cells reach the pluripotency state. But we don't see that in the neural trajectory and stromal trajectory. It goes down and stays low, relatively low. And importance of MYC for stem-like pluripotent cells were also experimentally validated. Right, so when we look at the top 20 interactions of the very early day one, actually day 2 is when they start to sequence, what we see is the enhancement of enrichment in pathways related to pluripotency, and also downregulation of epithelial mesenchymal transitions, which is the opposite of mesenchymal epithelial transition that cells are required to go through for pluripotency. So this also validates our insights. We look at a very small part of the regulatory network and also see that SOX2 and NANOG, their influence and their interactions with other transcription factors go up as we approach the pluripotency stage in the transitions. Lastly, we capture similar insights for mouse hematopoiesis, where we see that SPI1 is a driver that comes up for both the CMP and LMPP trajectories. But when we look at when we don't rank drivers just based on the gene itself, but look at the interactions as well, the SPI1 GATA1 becomes the top one, the top interaction for the CMP trajectory, whereas GATA3 doesn't appear in its top 10 at all. Lastly, the gold standard for basically understanding that we learned a good gene regulatory model is to test out perturbations. There is a published paper that performed gene knockouts for hundreds of genes in hematopoiesis. So if you look at the difference in how the relative population sizes change with knockouts, and we compare it to our results in a different data set, because of the growth term, we can also now learn whether the changes in the gene expression leads to apoptosis, so we incorporate that as well. And we see that while the numbers aren't exactly super correlated, the directions are very, very well correlated. Right, limitations. Of course, the transcriptomic data only provides one view of the cell state. So extending this to multiomic data would actually be very informative, not just only for regulatory interactions, but also when we look at lineage tracing data sets with multi-ohm data, we see that using both of the data resources actually increases the predictive power over progenitors by quite a lot. And we could also generate hypotheses on accessibility gene expression relationships by looking at the delayed Jacobian, so the partial derivative of the ODE after integrating it for a while. And that could give us now insights into potentially new interactions that we may not have in curated data sets. I'm excited about the potential of using this for understanding a cell's environment on its spatiotemporal trajectory, as we have spatiotemporal data sets now as well. I'd like to thank Caroline and Shiva for being supportive during this project. I also like to thank the members of Schmidt Center and Uhler Lab, especially Dora, who I had very useful discussions with on trajectory inference early on, and Chitian for those discussions on graph neural networks.
We have time for one question.
All right, thanks for the great talk. My question is, in the underlying physics that it's chemical reaction dealing with all these interactions, right? So that basically means it's always stochastic. Mm-hmm. And to my understanding that you're using an ODE model, which all stochasticity is in the initial state. And I do understand that, yeah, indeed, even if you use an SDE, you will still have a probabilistic flow ODE. But I guess once you start to use ODE and you admit that I admit there is stochasticity, how do you interpret the interaction, because you got the stochasticity absorbed into your velocity?
Yeah, so we actually don't know the shape of the noise in single cells, right? So with stochastic models, people make the assumption that there is Gaussian noise in all cells, and they place a Gaussian noise over the gene expression data of all cells. But that's probably not very biologically accurate. I would expect it to be more cell state dependent. And early versions, not for the trajectory inference part, but for the gene regulatory inference part, I did consider using SDEs. I don't see an accuracy increase.
I see. Thank you.
Yeah. Okay.